pleasure to introduce Jagath Reddy Junutula. Uh, Dr. Junutula has co-founded a NewCo that is currently in stealth mode. Prior to this, Jagath was at ModMab Therapeutics, Celerant Therapeutics, and Genentech, spanning 20 years. Jagath has been responsible in contributing to several INDs and IND-ready programs. His work focusing on drug discovery and development of biologics, including discovery and validation of antibody-drug conjugates, or ADCs, engineering next-generation ADCs, and development of bispecific T-cell engaging antibodies, has been widely recognized. He served as an R&D team member of an approved drug and one of the inventors of an approved polyvi ADCs. He developed a novel thiomab technology platform to generate site-specific ADCs to improve the therapeutic utility of antibodies. Jagath was a featured inventor in Greatest Hits in 20 years of Nature Biotechnology biomedical research. Jagath obtained his PhD in 1997 from the Indian Institute of Science, Bangalore, India, and did postdoctoral research at Stanford University. Jagath has authored greater than 60 peer-reviewed publications and is listed as an inventor on greater than 60 patents. He received a long-standing contributions to the ADC Therapeutic Field Recognition Award at the 2017 World ADC Summit in San Diego, California. Jagath serves as an editorial board member of leading journals such as MABS and ACS Medicinal Chemistry Letters. Today, he'll be presenting on engineering antibody-based designer drugs. Please take it away.
All right. Let's see. Hi, good afternoon, everyone. Sorry for the technical glitch. Thank you for the organizers from Precision in Drug Discovery Preclinical Virtual Summit to inviting me, especially Sherry. And it's my pleasure to talk about engineering antibody-based precision medicines. Before going to the actual talk, I like to show something personal, actually. So these two cartoons are hand-drawn by my children, actually. The first one is antibody-drug conjugate. That's a site-specific antibody-drug conjugates. My son drew this is about 8 years ago when he was like 9 years old. And there was an important milestone happened there. I wouldn't say it's an interesting one for me personally. So that was a site-specific antibody-drug conjugate that I developed at Genentech. It came in Nature Biotech and all that. But for strange reasons, unfortunately, the program shut down, which I can't talk about reasons. So then second one is hand-drawn by my daughter, actually, 4 years ago. This is a T-cell recruiting bispecific antibody. So this is the work I have done, started working at Genentech in 2010 and went on to do till 2014 and fantastic work. But then I decided to leave, even though work was going extremely well for reasons to fix the first one. And so my daughter thought she will draw. And every day I see these two pictures in my office. And they remind me I need to do something special to bring them back. So let me tell the story, what I've been up to. So today, actually, I will not speak on antibody-drug conjugates. I already spoke about it about a week ago in a different conference, and it is the entire talk is on my LinkedIn. Anyone is interested, you're most welcome to go ahead and listen to it. But today, I'll speak about T-cell recruiting bispecific antibodies. My overview of the talk would be introduction to antibody-based precision medicine and T-cell engagers, and what are the T-cell dependent bispecific antibody program that we worked at Genentech, and that led to starting a new project, which I oversaw, that's called Peptide HLA project. That further led to a collaboration with Immunocore, a company based in UK, that I oversee completely that entire platform collaboration I oversee as a research lead. And that led to some important lessons, actually, how the industry is looking at these molecules recently. It's very interesting lesson. Personally, I learned, that's my personal perspective I'm going to share. On current status of T-cell engager, where do we stand and where do we go from here? A quick summary of antibody-based precision medicines, what we are today. So-called targeted cancer immunotherapeutics. Everyone knows about function-blocking antibodies such as Herceptin, Rituxan, Avastin, anti-PD-L1. These are the targets meant for targeting antibodies, meant for targeting cancer cells, and they work beautifully. Then we have antibody-drug conjugates, such as Adcetris, Kadcyla approved, and then new ADCs. There are almost 180 went into clinic so far. Then we have other spectrum side where we have antibodies that recruit immune cells into the cancer and eliminate it. The one of the class is the bispecific antibody, today's topic of interest, which I'm going to talk about it. And then there are other approaches too, activating immune systems such as immune checkpoint inhibitors such as anti-PD-1, anti-CTLA-4. There are also other approaches such as ImmTACs. I'm going to cover this one also. But then a lot of hype happened past few years on CAR-Ts, NK cells, and all those. I will briefly touch on all those things too, actually. So before going to the topic, I'll just quickly summarize what is the current status of antibody-drug conjugates today. Again, as I said, I gave this talk a week ago. It's on my LinkedIn. But just quickly summarize, antibody-drug conjugates, they're doing extremely well to me personally, because you got 10 approved antibody ADCs to date. 183 went into clinic. 100 of them still active. 80 of them discontinued. Well, over 929 clinical trials actually happened. Thanks to my colleagues of Anson Wade, I have been working with them for a number of years for sharing this important data set. What you see here, there is a lot of activity since 2010 in ADC field. But what is bothering me personally, or my colleagues in the ADC field, failures. 90% of the failure rate. So I know ADC researchers have been working very hard, but there is huge unmet need here. Not necessarily the 100 which are active are going to all of them will be active too. I mean, all of them get approved too. Probably another 10% will be successful. But why are we dealing with this high failure rate? High failure rate in the clinic means cost will go up for approved drugs automatically. So can we actually do something else to cut down these high failure rates? I think that is something very close to me and dear to me. I'm seriously looking into that. So let's switch a gear towards T-cell dependent bispecifics. What are T-cell dependent bispecifics, by the way? Very simple. You have an antibody, one arm binding to a cancer cell, another arm binding to a T-cell. So this antibody sort of acts like a bridge between cancer cell and T-cell, and the T-cell is being recruited to cancer cell, and T-cell gets activated, and then activated T-cell releases porphyrin granzymes, and they will make spores into this cancer cell and kill them. I just wanted to make sure everyone can hear me, because I see a blank screen here. Can you guys hear me?
Yes.
All right, thank you. Then there's another approach, very cleverly approach, a company called NewCo has done. They do the same thing, but they use TCRs actually for that. Recruit the TCR. They use TCR fusion protein to recruit the T cell to the cancer cell. And this targets a different class of targets. They call HLA peptides. Very interesting story. I'm going to cover on this very detail later. So let's go back to the history of the bispecific antibodies. 1996, my former colleagues, Len Preston and Paul Cotter at Genentech with John. I know all of them personally. And I worked with Len and I worked with Paul also. Beautiful minds, I would call them actually. Think about 1996, they thought about a bispecific antibody, and today we are in 2021, actually. Okay? I mean, you have to go back and think to that era and see how they thought about this. Beautiful idea. They thought about making knob and holes in the FC and express them only knob and hole come together, not knob or hole. Beautiful concept. Today's success of entire bispecific technology depends on this interesting idea. By the way, this IP is no longer there. Anyone can use this IP, by the way. But when I think about going back to 1996, that time humanization just happened. That's again thanks to Len Preston and Paul Cotter. Fantastic story. But then field moved on ever since. Hundreds of different farmers. Everyone uses their own format. Which one to use? Which one is good? Which is safer? Which is clear? Which clears faster? Which distributes better? Which is immunogenic? Which one makes CMC issues easier? So many questions. Everyone says mine is best. Yeah, sure. Many different formats are doing extremely, especially past 5 years, there's huge improvement ever since. It started with the blinatumomab with Micromet. So I think a lot of credit goes to blinatumomab Micromet. It's a small company in Germany, and eventually that was acquired by Amgen. And what they have done, they actually made SEFE fusion diabody. One side binding to CD3, other side binding CD19. I guess these are the people should get highest credit for making this concept real. That's why I believe small companies actually bring these interesting technologies to the industry. So small biotech actually a huge role to play in bringing novel technologies. Because big pharma biotech, they never want to take risks. But small companies do take risks, actually. That's what I like about small companies are being very aggressive, very passionate, and trying out new technology. And this is one of the story, Micromet story. Fantastic story. And they made it till to date. This is the only approved drug in the bispecific T-cell recruiting bispecific antibodies. This was done a while ago. But what's going on in the field? Why it took so long to fill the gap? So my story at the T-cell dependent bispecific antibody program started at Genentech in 2010. Alan Evans, was the director of this program, asked me, Jagath, could you join us to take on some of the projects? And Sunil, my colleague who worked with me, and we together took on some of the challenges that Alan team had, and Alan team already made fantastic progress in this group. I'm going to quickly summarize some of the things Alan's group has done. To cut long story short, I want to tell about comparison between ADCs, T-cell engager, TCR mimetics, CAR-Ts. Everyone talks about these four things. They're all precision medicines, antibody-based precision medicines. What is the target selection criteria for all of them? For all of them, a target has to be a cell surface protein. Except TCR mimetics, a protein could be on the intracellular, but eventually it comes either in the form of HLA peptide on the surface. Copy number. This is actually the most important feature for all these targets. ADCs. You cannot afford to target an ADC target which is less than 10,000 copies. Someone says HLA peptide they want to use as an ADC, have a good luck. Don't even try, please. HLA peptide copy number is only 100. T-cell engagers. Yes, they work. They can work up to 1,000 and above copies. It all depends on the affinity of the T-cell engager. If you have femtomolar, you can go up to even 10 copies. If you go picomolar, maybe you need about 100 and more than that. If you go nanomolar, you probably need more than 1,000 and above. So this depends on how clean is your target is. TCR mimetics, beautifully shown by Immunocore and team, you need copy number above 10. That's good enough. And CAR-Ts, people are shown even one. Copy number of 1, also it works. Good luck. So internalization, target internalization is not needed at all for all this. Except ADCs, you need target internalization because ADC has to get inside the cell, they get degraded, payload releases, that's fine. But rest all, you don't need target internalization. Doesn't matter whether it internalizes Ideally, you do not want targeted internalization for the rest of them. So in other words, you could choose targets. They don't work for ADC, beautifully work for the rest of the three things. Normal tissue expression. This is a big deal, actually. ADCs can get away having moderate tissue expression in the targets because the copy number below 10,000 anyway doesn't work. So therefore, you can easily identify a target that abundantly express in the tumor, minimally express in the normal. You can get away easily with ADCs with a good window. Let's come back to T-cell engager. T-cell engager, you could still get away, but you still need to make sure the expression is very minimal in the normal tissues. However, you could actually tune the affinities. Let's make nanomolar affinity, not femtomolar affinity. Then you can get away with weak expression. TCR membradics, you got to be super clean. No good luck otherwise. 10 copies. That's good enough to kill the cell. So it has to be super clean. No excuses here. CAR-Ts, yes, no excuses. So now you know where you're going. We are going, if you have a target that's super clean, super duper clean, you can go with TCR membradics and CAR-Ts. If you have moderate, abundant expression, but moderate expression is normal, you got to go with ADCs. Somewhere in between, you go with T-cell engager. Actually, you know what? They don't compete with each other, all these programs. They can nicely synergize with each other. That's what I believed in, and that is true, actually. I worked with all these programs past 20 years, each one of them. I know the rules very well. So this is actually a program developed, overseen by Alan Eben when he was Genentech. He calls T-cell dependent bispecifics. Beautiful story, actually. 30 people were involved. I was fortunate to be one of them in this team. And I've seen from beginning to the end of the molecule development, how they developed CD3 antibodies and target antibodies, no big deal because Genentech has been making these antibodies for 20 years. So, but they made their own CD3 antibodies and assembled it. Fantastic protein chemistry team. Fantastic job. This is all published. You can go read it. So this is actually a very interesting, this is the first proof of concept data was published by Alan Eben's team at Genentech. Fantastic story, Science Translational Medicine, published in 2015. Beautiful data. Laura Sun is the lead author who major contributor for this program, actually. So what they have done, they made a CD20, CD3 bispecific. Works beautifully. Wherever target is there, it works. When there is no target, no, it doesn't work. When there is no CD3, it doesn't work. Multiple cell lines they tested. They tested in CNOs. Everywhere they have done. Beautiful NHP studies. Everything is there in this paper. You can look at it. Fantastic data. I think this program is in phase 2 now. So what me and Sunil actually did while Alan and the rest of the team working on many different targets. So me and Sunil actually picked a number of targets, almost 10 different solid tumor targets. Alan was focused on hem-onc targets, and me and some other people were focused on solid tumor targets. We narrowed down a couple of targets. Beautiful stories were there. But, for me, time was ripe, and I had to leave for different reasons. Nothing to do with science, but I decided to take on something else, a challenge, and therefore I had to leave halfway through, and we couldn't have completed stories. We couldn't publish. No one knew what I've done. But also what Alan actually challenged me while I was working with him, Jagath, could you take on something else? I know we have worked on tumor cell surface proteins. We are pretty much done at Genentech. Every cell surface interesting protein was made antibodies and all that, but we need to expand our target landscape. So could you work on HLA peptides? HLA peptides actually is a very interesting story, actually. So I took on that challenge, partnered with 3 of the outstanding scientists at Genentech, Jermaine Fu, Vivian Lee, and Sailor Panoski. Fantastic individuals. So we worked on actually 18 months. What is HLA peptides? Everyone knows. Its other name is MHCs. So how the HLA peptides work? HLA peptide typically is a complex with alpha beta chain of MHC or HLA, complex with the peptide that recognized by TCR, T-cell receptor. T-cell receptor is simply mimics like antibody light chain and heavy chain. You can see your alpha and beta chains. They mimic CDR. They have all CDRs. If you put antibody Fab and TCR extracellular domain, they actually can overlap very well. The interesting thing about is you have a 9 amino acid peptide sandwiched between HLA peptide, HLA and TCR. So these 9 amino acids contribute for this junction. In other words, there are about 4 amino acids occupied by HLA, about 5 amino acids occupied by T cell receptor. Period. You're dealing with specificity with that less number. So now if you want to make antibody against HLA peptides, you have to mimic the TCR essentially. In other words, you will be identifying an antibody that binds to 3 to 4 amino acids. That's it. You see that specificity is super duper important here. We went on to do that actually. We went on to do for 2 targets, NYESO and very famous HLA peptide, and then MAGE-A3. So we actually shortlisted these 2 and decided to focus on MAGE-A3. We spent 18 months. Seriously. We got actually first antibody in 6 months. We got jumping around. Then we looked at, it was specific, but we were not satisfied. We went and looked at the entire proteome we searched with that peptide, 9 amino acid peptide. Then we got hundreds of different matches we got. We synthesized all the 200 peptides we synthesized and screened. Guess what? Almost 70% of them did not bind, but 30% bound. Actually, I looked at the papers prior to 2011, which are published. They just show specificity with only 1 or 2 peptides. They're all actually garbage. You don't believe. Anything HLA peptide antibody people made prior to 2012, just don't believe it. Because they have not looked at entire proteome. They looked at, except only one company, NewCo, has done this. They look at the specificity. They look at everything, entire proteome. Except Immunocore, Adaptimmune, anyone else published prior to 2012, I looked at personally. They have not shown comprehensive specificity analysis. So when we did 200 peptide analysis, the entire proteome covering, we found was shocking. 30% of them actually bind it. So that means they're not specific. Then Jermaine and team went back and re-engineered it. We narrowed down to 30 to 10. We went third round, we narrowed down to 1. So it took us 18 months. Meanwhile, I went to Europe in 2012. I heard Bent Jacobson, a co-founder of Immunocore and Adaptimmune in Oxford. So I heard his talk in the Antibody Engineering Conference in Europe. I was so fascinated about it. I didn't get a chance to talk to him at that time. I came back to Genentech. I talked to Alan. Alan, what are we wasting our time? 18 months we took to make 1 antibody for 1 HLA peptide. Here you have a company, they already figured it out. 10 years ago, they started. Then Alan said, let's go present it to the senior management. Senior management approved it, go and have the discussion with them. We started in January, early discussion. By end of March, April, we actually signed the deal with them. I think that's, to my knowledge, fastest deal ever happened in Genentech at that time. Because before that, Genentech was always focusing on internal programs, not external programs. So we are proud of doing this. But why we are so excited about Immunocore and Adaptimmune? What is so special about it? Again, Bent is a mastermind, and his right-hand Namir, I like him. He's a good friend of me, actually, now. Fantastic scientific mind, actually. So what did we like about it? Here is a snapshot about IMTAC. They call IMTACs, immune mobilizing monoclonal TCRs against cancer. What do the IMTAC do for actually? So typically you have ADCs, CAR T cell, bispecific, they all actually target the typical cell surface proteins. What do IMTACs do? They target actually HLA peptides. Where these HLA peptides coming? You have every protein inside the cell, they undergo degradation. Is cell surface or cytosolic, doesn't matter. They go degradation through proteasome, and the proteasome presents these peptides on HLA, and the HLA peptide comes to the surface and lives there for maybe a few days and goes back. So this TCR, T cell receptor mimetics, basically, I engineer them to bind this HLA peptide. What they have done cleverly, they used an antibody against CD3 and they fused it. Now you have activated TCR, essentially. Just like T cell dependent bispecific. Beautiful concept. So as you can see here, what they have done, everyone knows TCRs is a weak binders. They are micromolar affinity. So you cannot use as a therapeutic. So what you have to do, you have to engineer the TCR. And they have done this in 2000s. Like 10 years ago, anyone thought about it. That's Bent is the mastermind behind that. So they engineered, they made PH display library, the beautiful paper they published actually. They engineered, they got femtomolar affinities. Really, I'm not joking, it's femtomolar, picomolar, femtomolar, okay? Picomolar affinity they identified. But in addition, they couldn't use TCR ECD as is, they fall apart. They have to engineer a disulfide bond to stabilize the TCR. So again, they have done a lot of CMC work on that. So this is long story short, it's a Nature Medicine paper they published in 2012. Beautiful paper. How they went about engineered the syntax and they showed the specificity, how the affinity actually varies the potency of the molecule, from left to right. As you decrease the affinity, it decreases the potency. Nice data. You can go read it. Again, one of the best paper I've read it past 20 years, I must say. So what did they do? I mean, they took this molecule. By the time when we interacted with them, they completed the phase 1 with their lead program, GP120. And I think it's going to be very close to approval now. So if I'm correct. But what do they do basically? Again, as I said, you are actually now thinking about targets, not cell surface target. They are actually cytosolic proteins. There's so many cytosolic proteins abundantly expressed in the tumor and no expression in the normal tissue. Now you're thinking about the cleanest possible target that you can think about in the proteome. Okay. But unfortunately, they only express in copies number of 100. That's it. To get target copy number of 100, nanomolar affinity is not enough. You have to go picomolar affinity. That's what they have done. They went ahead and get single digit picomolar. They got it. And these are specific. They bind and they act because you have a CD3 binder. They bring the T cells to the tumor surface just like TDBs. And they activate the tumor cell, T cell, and activated T cell kills the tumor cell. Again, same concept. But why it is so special? Now you are going a different target proteome. 80%. Typical cell surface receptor, only 20%. Now you are covering 80% of the protein targets. Completely new target landscape. No one explored it. That's the beauty of this company. Of course, they didn't have a easy ride. When we are evaluating back in 2013, they had their sister company, which is Adaptimmune. That is a T-cell therapy company. And Immunocore is a biologic company. And Adaptimmune is a T-cell therapy company. Adaptimmune has a very bumpy road. First program, their MAGE-A3, 2 patients died. Literally. Everyone knows, right? T-cell therapy company, how they do their science. You cannot do preclinical studies because there is no equivalent of a monkey molecule. So what do? They make construct, take it to the clinic. So it's basically short timelines. So what FDA and the equivalent European agency they've done phase 0. In other words, you can inject this cell therapy molecule to only 3 patients sequentially. So Adaptimmune hit a roadblock. They actually 2 patients died in the first clinical. They had to halt the program. Even Immunocore has to halt their MAGE-A3 program because of that. But they didn't give up. That is what I liked about their approach. What they have done, they went and nailed down why actually it killed the patients. They nail it down to the molecular level. You read these 2 papers, fantastic stories. I wish all the CAR-T companies follow these people actually. Patients die, they move on to next molecule without even trying to understand science behind why patient died it. But Bent and team went on to figure it out why they died. Because the MAGE-A3 TCR, what happened is it was cross-reacting to tidine. Tidine is expressed in cardiomyocytes. But they checked cardiomyocytes, actually adult cardiomyocytes, they checked it did not cross-react. They had to look at stem cell progenitor cells of cardiomyocytes to identify the issue. So based on this story, They did a comprehensive safety analysis. They figured out. This is PLO's paper they just published recently. What they have done, every possible human tissue, they test their immunocore molecule or adaptive immune molecule before going to the clinic. I don't think any of the CAR T companies are doing this. Better they learn from them. At least we can avoid killing the patients. Fantastic story. A lot of people don't read. These are, PLS, ordinary journals, right? No one wants to read this ordinary journal. Everyone wants to read Nature Cell Science. That's all. No, please go back and read these papers. Super important paper these are actually. These papers can save patient lives. And this is what excited us as a Genentech when you're doing diligence. This is the company we want to work with. We signed deal in 3.5 months. We are the first one to sign deal with Immunocore. They were talking to a big pharma for 2 years. They didn't sign it. As soon as we signed the deal, 2 other companies signed deal with that. Because this is the best scientific mind, scientists they figured out, nailed down. This is what you would want to see in the companies, basically. Not trying to make money and exit companies and make few people billionaires and move on with it. That's what is going on. Poor ethics in the biotech industry, unfortunately, these days. Everyone wants to make money and move on. Exit, exit, exit. No one is looking after patients. So what's going on with the rest of the T-cell engager technologies? I think these papers, recent papers, beautiful stories, actually. It caught my attention. Here, a lot of people are excited about CAR T, right? What would antibody cannot do, CAR T can do, actually? CAR T is, you have to admit, they have done fantastic technology they have done. They brought in T-cell activation, signal 1 and signal 2. Unfortunately, T-cell dependent bispecific never caught up with that. Signal 2 is very important because you need to have prolonged T-cell activation and you have to have durability and all that. So that's what CAR T has done, actually. 20 years ago, they have done this. I don't know why T-cell dependent bispecific didn't learn that trick. Finally, someone opened their eyes. 2019. Fantastic paper. So when they combine CD3 with CD28, beautiful activity. This should have been done 15 years ago, if you ask me. I was not in the field. But this is the data you need. So in other words, this molecule can beat the CAR T any given day. Yes, I'm looking forward to this molecule one day in the clinic. This is another one, which is my former Genentech colleagues. Fantastic data. What they have done, there is cytokine syndrome, release syndrome. Everyone faces. CAR T face this problem. T-cell dependent bispecific face this problem. Everyone, T-cell engager, TCR, everyone has this problem. When you inject it, you are going to have cytokine storm. How are you going to take care of it? So what they have done, actually, they came up with TNF-alpha cytokines. IL-6 actually are the contributors for this. So they knocked down TNF-alpha. T-cell activation is not compromised. So what they did, beautiful data, actually, team and colleagues. I loved this paper, every bit of it. So he used anti-TNF-alpha, anti-IL-6 and suppressed the cytokine release and without compromising the T-cell activation. Hey, actually, this is fantastic. You can actually do it. So what's going on with the T-cell engager clinical pipeline status? Just a few minutes I want to spend on this. A lot of activities going on. Heavy activity in phase 1, actually, if you look at. Thanks to my Hansen Wade colleagues for sharing these database slides. Almost 312 trials for CD3 targeting T-cell engager. Everyone is going after. Is there anything interesting in this? I don't know. Because everyone is copying each other. They just simply either making a different format, different target, a different thing. Believe me, they are all going to have the same problem. Cytokine release syndrome. I don't know how they are going to have window. I'm not sure. I'm not convinced as a scientist. Look at this activity past 5 years. So much activity. But believe me, T-cell engagers actually didn't get as much deserved attention as CAR Ts. I don't know why CAR Ts get so much attention. I don't know personally. Again, I'm not against the beautiful technologies, but antibody any given day can do better than a cell therapy company.
And Dr. Jagath, just a soft reminder, think we are closing in on the next presentation.
All right, I'm almost there. So these are the targets which everyone uses. Same targets, multiple times, 10, 15 times. This is what I was telling about challenges with the T-cell engagers. You have CD3 only activation, signal 1. Off-life either too short or too long. Cytokine release syndrome is the biggest issue. Getting back to the summary slide, where, believe me, everyone uses all these things. People are thinking about combinations and all that. Believe me, who is going to pay for that? Combination therapies. Patient? Each drug costing 200,000, 100,000. Healthcare is going to be crippled. You got to think something different. You can't think about combination therapy. Believe me, patient cannot afford it. So I've been thinking very hard. Finally, I found my team that I want to work with to make a difference for these ADCs, T-cell engagers, and beyond. I found my outstanding partners, antibody engineering team, one of the best in the world, Dr. Dev Sidhu, and his colleagues. We are doing something different. So hopefully, I get to tell my story in coming months. With that, thank you. Thanks to all my passionate bispecific antibody colleagues in the pharma biotech for taking challenging problem. Thanks to all these animals. I wish all the CAR-T companies can use this. They can't. But think about Immunocore, Adaptimmune, what they've done. It fantastic story. At least think about doing those in vitro comprehensive safety studies before taking to the clinic. Patient lives can be saved. Okay? With that, thank you all. Thanks to all the patients for taking the risk and for making few individuals super rich. Thank you.
Thank you so much, Dr. Jagath. If anybody has any questions, please email us. We can connect you with him. You can also connect with him on LinkedIn as well. Yeah, thank you so much again.
Thank you.
So we'll be going to our next presentation by Dr. Woody Sherman. He is the Chief Computational Officer of Roivant Sciences. So I'll just be transferring you all right now.